Next instrumentation methods is the spectroscopic method. So it is based on the measurement of the interaction between the electromagnetic radiation and the analyte atoms of the molecules. So kani ato makita diri sa slide is an example of a spectroscopic method. So the use of UV vis to measure the concentration of the solution by the amount of light absorbed by the substance across UV visible nga mga ranges. So for example, if you are tasked to determine the concentration of iron in your water, so we can use the UV vis para ma-determine nato kung pila yung concentration. And then next is the other nga mga instrument nga gina-use sa quantitative analysis, the flame photometry, the mass spec, chromatography for separation and then identification of components nga naa sa unknown mixture, and then electrophoresis for proteins, and then other nga mga type of instrument. So we have here HPLC, so complicated nga instrument used to separate, identify, and quantify nga naa sa mixture. So basic analytical process. So in analytical lab, naatay gina-follow nga processes, systematic process para mag-carry out og experiment or analysis. So first is to select a method. So you will consider here your level of accuracy, the number of samples nga inyong i-analyze, then kung unsa ka complex ang sample or pila man ang components nga dapat ninyong i-analyze in that sample. And then afterwards nga nakaselect na mo og method, then you will acquire the sample. So you will have your sampling to get your sample. And then afterwards, if naa na mo'y na-acquire nga sample, you will process the sample. So you will prepare your sample for laboratory nga mga analysis. So preparing your laboratory sample, you will define your replicate here, and then you will also prepare solution para sa laboratory nga mga processes. Then we need to eliminate interferences. So kinsa man ni silang mga interferences? Kaning mga interferences is the species that causes error by enhancing the quantity being measured. So we will select reagents or methods para ma-minimize ang mga interferences. And then we will calibrate our instrument. So before na to i-inject dito atong sample sa instrument, we will have to calibrate to check if ang ato ang mga instrument kay gahatag pa ba siya og correct nga mga value or sensitive pa ba siya nga instrument. And then if naanatay data nga gi-generate from the instrument, then we will calculate the instrument. So the computations are based on the raw data collected in the measurement steps. So characteristic of a measurement, the instrument, stoichiometric of the analytical reaction. So if we have now the result, so we will also evaluate kung unsa ang pasabot sa katong nga result. Taas ba siya or mubo ba siya or unsa bay nahitabo sa atong result. So you will evaluate, the experimenter must provide some measures of uncertainties associated with the computed result if the data are to have any value. So we have here the types of sample. So qualitative, again, determination of the identity of the chemical species, and then quantitative for determination of the relative amount of chemical species. So qualitative, for example, we have color changes nga nahitabo, and then quantitative, for example, is your titration. And then we define analytical chemistry based on our sample size. So we have macroanalysis, semi-microanalysis, microanalysis, and ultra-microanalysis. So for macroanalysis, so the amount of analyte is more than 0.10 grams or 100 milligram. So for example, if we suspect pollutant in 1 gram nga soil sample, then we can say nga that specific analysis is a macroanalysis. Dinagko. And then semi-microanalysis, so the amount of analyte is between 0.010 to 0.10 grams. So for example, if we were tasked to determine the amount of a drug in 5 mg nga sample problem, so we will determination of glucose in a blood sample. So is an example of semi-microanalysis. The microanalysis naman, so the amount of analyte is 10 to the power of 4 to 10 to the power of negative 2 grams. So example is the determination of your creatinine in your urine sample. Ultramicroanalysis, so amount of analyte is less than 10 to the negative 4 grams. So determination of arsenic or boron or nickel or any other nga mga metals nga present sa kaning blood or urine. And then based on the analyte naman, we have major constituents, minor constituents, trace, and the ultra trace. So for major, the amount of analyte is 1 to 100% of the population. And then minor constituents, 0.1 to 1% of the population. And then trace constituent, usually mga ilahang concentration is 1 ppm or 1 ppb. And then ultra trace, the amount of analyte is below 1 ppb or parts per billion or parts per trillion. And then sampling. So in analytical chemistry, sampling is one of the katong steps sa atong analytical process. So unsa ginabuhat sa sampling? So a process of collecting a small mass of materials na ang iyang composition nagrepresent sa bulk of the material being sampled. So it is the most difficult aspect of the analysis. So samples are analyzed, but species or concentrations are determined. So we have real sample. So analysis of the real sample are complicated because of the presence of the sample matrix. So example, sampling of a human blood for determination of blood gases illustrates the difficulty of acquiring representative sample from a complex biological system. So the concentration of oxygen and carbon dioxide depends on the different variables. So by applying a tourniquet incorrectly or hand flexing by the patient, this may cause the blood oxygen concentration to fluctuate. So mao nang some other kay naay mga techniques in withdrawing mga blood samples. So the goal of the sampling is to ensure that the item chosen kay representative siya atong bulk of the material. And then we have gross sample. So these are the representative sample that are collected from the source. So katong tanan ninyo nga na-collect nga mga sample from your sampling site, tawag na to sila og gross sample. And then we have the laboratory sample. So these are samples that are reduced in size and being homogenized so that they are measurable in the lab. So katong gross sample ninyo nga gi-collect sa inyong sampling site, if atoan siyang ma-process sa lab, atoan nang na-reduce ang iyahang size or atoan na siyang na homogenize, so we can say na that specific sample is for laboratory samples. And then this will be your assignment. So in 1/2 crosswise, you will draw a Venn diagram and then follow the instructions na given, and then you will compare and contrast the characteristic of the different analyses in analytical chemistry. And then at the back of your paper, you will answer the following questions. So sa atubangan is kaning Venn diagram, and then sa likod is your kaning question. So this will be by pair.